Good evening, ladies and gentlemen. On behalf of the Royal Academy of Engineering, may I welcome you all most warmly to the 2007 annual lecture of the UK Focus for Biomedical Engineering. This is the third in the series of these lectures, and I'm delighted that our first lecturer, Sir Christopher O'Donnell, is in the audience today, so I'm very pleased that he's able to join us. I'm Tony Unsworth, and I'm Director of the Centre for Biomedical Engineering at Durham University, and I'm the current Chairman of the UK Focus for Biomedical Engineering. And we are indeed very fortunate this evening to welcome Dr. Leroy Hood, whose name is synonymous with the subject of tonight's lecture, systems biology. Many of you will be aware of the joint report, a copy of which I have to wave at you here, the joint report which was published in February, the Academy of Medical Sciences and the Royal Academy of Engineering. And as you see, this is the Systems Biology, a Vision for Engineering and Medicine. That was published, as I say, in February of this year, and it describes the iterative application of biomedical knowledge with mathematical, computational and engineering techniques to build and test highly complex models of biological systems. And this evening's lecture is therefore a natural progression from this report towards the implementation of the outcomes. And who better to lead us on that journey than Dr. Hood? But before I do a formal introduction, I have a few housekeeping issues to mention. First of all, should a fire alarm sound, and we're not expecting a fire alarm to sound, but should one sound, you can see that there are exits to my right and to my left, the little green signs. There are also exits on that side, two of them at the far corner over there. When you leave through any of these doors, you'll finish up in the foyer just by where you've been having your coffee. I'm told that you then have to go out into the street, into Wimpole Street, turn left when you go through the main door, and head towards the chapel or the church, which is right in front of you as you turn left. And that's where you must wait for further instructions. We hope that we don't have a fire alarm, but that's what you do. Could I also mention, please, that don't leave your mobile phones on silent, because if you do, and you do get a call, they do get picked up on the system here. So we wouldn't like that to happen in the middle of the lecture. So please shut them off before we start. I'd also like to thank Connor Ward, particularly of Lovells, or the IP lawyers, because they have sponsored this lecture over the last 3 years. And I'm very grateful to them for their generosity. So a thank you to Lovells for that. Now, Dr. Leroy Hood is president of the Institute of Systems Biology in Seattle. He obtained his MD from Johns Hopkins School of Medicine in 1964, and his PhD in biochemistry from California Institute of Technology 4 years later. His first professional appointment was at Caltech, and whilst there, he and his colleagues developed 4 instruments for DNA and protein sequencing and synthesizing, which revolutionized genomics by facilitating rapid automated sequencing of DNA. These instruments laid the foundations for modern molecular biology and genomics. He applied these techniques to diverse fields such as immunology, neurobiology, cancer biology, and systems medicine. And it was in the late 1980s that Dr. Hood realized that systems biology approach was the way forward. In 1992, he moved to the University of Washington. It was here that he developed the 5th instrument, the inkjet oligonucleotide synthesizer, which synthesized DNA chips and initiated systems studies on cancer biology and prion disease. In 2000, he co-founded the Institute for Systems Biology in Seattle, an institute of world renown today. He was awarded the Lasker Prize in 1987 for studies on immune diversity, the 2002 Kyoto Prize for developing the 5 instruments I've already referred to, 2003 Lemelson MIT Prize for Innovation and Invention for his DNA sequencer, the prestigious 2004 Biotechnology Heritage Award and the Association for Molecular Pathology Award for Excellence, as well as the 2006 Heinz Award for Extraordinary Breakthroughs in Biomedical Science at the genetic level. And this year, he was elected to the Inventors Hall of Fame. He holds no less than 14 honorary degrees, has published over 600 papers, and holds 14 patents. Dr. Hood not only is a member of the National Academy of Sciences, but also the American Philosophical Society, the American Association of Arts and Sciences, the Institute of Medicine, and the National Academy of Engineering, as well as founding 14 biotechnology companies. Well, in the interests of leaving some time left for the lecture, I've abbreviated this introduction, but it must be clear from what I've already said that we can look forward to a very special presentation this evening, and I now call upon Dr. Hood to deliver the 2007 annual lecture of the UK Focus for Biomedical Engineering on systems biology and medicine.
Well, thank you very much. Where I'd like to end up at the end of this lecture is to tell you about the revolution I see coming in medicine, where we'll move from a reactive type of medicine to one that's predictive, personalized, preventive, and participatory. And I'll explain what all of that means. And I thought the best way to do it would be in the context of a career now that's spanned more than 40 years, and in a sense, as I grew up with the biotech industry. And I've participated in a number of paradigm changes in biology, and we can argue about what a paradigm change means. But what I'm going to talk about are some of these paradigm changes in the context they created then for thinking about systems biology and systems medicine. So these include bringing engineering to biology in terms of technology development, but not just technology development, integrated technology development, high-throughput technology development, thinking about the Human Genome Project, thinking about the creation of a cross-disciplinary environment for doing biology, and finally systems biology and systems medicine. And I think one of the things that really has been striking to me is in almost every case where there was really a new idea like this, it only achieved realization through the creation of new organizations. I think one of the fundamental problems that we have in our society today is old organizations can't do new things. So we can think about creative ways that maybe in the future old organizations actually could do new kinds of things. When I started at Caltech in 1970, I told the chair that I want to spend half my time doing molecular immunology, half my time doing technology development, and he didn't say anything at the time, but what was really interesting is once I got going, 3 years later, the chairman, Bob Sinsheimer, came into my office and said, I'd like to advise you in the strongest possible terms to give up this technology development. We really want you to focus on your biology. And of course, I disagreed with that. And 25 years later, had the presence of mind to ask him why in the world he'd ever said that. And he said it because the senior biologists in the division of biology, apparently to a person, felt it was unseemly to have engineering and biology. And what they'd really insisted is, why doesn't he just move me into engineering? And he said, at least I didn't do that. I'll say parenthetically, I got tenure 2 months after he came into my office. So at 3 years, that's really early for tenure. So the issue didn't really have to do with tenure. The issue had to do with taste. And that same taste is the reason I ended up leaving Caltech a little bit later, as you'll see. So when I went to Caltech, my thinking about technology development was really predicated on two ideas. The first idea was that the frontier problems of biology have to drive and dictate the nature of the technology you develop. And once you've developed a new technology, then that potentially has the opportunity to revolutionize the biology. And the automated DNA sequencer, I think, was a nice example of that. The second idea was that I was really interested in and beginning to think about biology as an information science even then, and interested in getting instruments to decipher biological information. And at that time, we knew about DNA and RNA and proteins, so we thought about developing then instruments that could decipher, and we can talk in a few moments about what exactly we mean by decipher. But as you've heard, over the years, we developed these 5 different instruments. And if you think back about those types of biological information, they do indeed let you decipher many of those things. What was interesting was in about 1979, I had a friend come to me and say, why don't you think about commercializing the instruments you're developing to make them generally available to the community? And I thought this was a terrific idea, so I went to the president of Caltech, Murph Goldberger, and said, hey, how about commercializing these technologies we're developing? And his attitude was classic for the times. He said, the role of an academic institution is scholarship and education. It has nothing to do with commercialization. If you want to do that, go do it yourself. So being young and enthusiastic, I did. I went out to 19 instrument companies over the next year and was totally unsuccessful in persuading any of them to take on this assignment. So I then got a call from Bill Bowes, a very famous San Francisco venture capitalist, who said, look, I hear you've unsuccessfully shopped these things around. I'll give you $2 million to start a company. And so I said, gee, this sounds really terrific. So I went back to Murph Goldberger, and Murph was really negative about venture money because at that time, Harvard was going through this affair with the Genetics Institute, and was venture capital money dirty money, which is what Murph claimed, or was it in fact? But what persuaded him was that there were no other options. So we were almost ready to sign this agreement with the venture company to start the company that was Applied Biosystems, became a leader in molecular instrumentation later on, when I gave a lecture to the Caltech trustees, and among those trustees was Arnold Beckman. And I'd gone to Beckman Instruments 3 different times, and the 3rd time they told me not to come back. They really understood what I was shopping, and they weren't interested. So Arnold came up after this lecture about the vision of what these instruments could do for biology, and he said, this is fantastic. This is exactly what Beckman Instruments needs. And I said, well, you know, I wasn't so sure they were interested. And then ensued a very complex, awkward situation, which I won't discuss. But I will say we did sign the venture agreement. Applied Biosystems got started, and it was enormously successful. And from that, I learned 2 important lessons. So the first lesson is, if you have big new ideas and visions, you go to the top. You don't go to middle-level people who are interested in profit and loss. The second was, it was incredibly fortunate. None of those instrument companies took me up on my offer, because none could have hired the talent, could have had the focus, and could have put all the effort into getting those instruments out that Applied Biosystems did. So it was really the first example of a new organization really being necessary for the commercialization. So I did start Applied Biosystems, and then the second company I started was another successful one, Amgen, as you heard. Went on to start a number of other companies, and the idea was really, I've always felt that scientists have an obligation to society to transfer knowledge, and that's a different way of looking at it than dealing with the commercial side of things. The automated DNA sequencer, which we were really starting to make progress with in 84, 85, got me into the next paradigm change, and that was the Human Genome Project. So in 1985, it was this really audacious, can we sequence 3 billion nucleotides from the human genome? And Bob Sinsheimer, who had become Chancellor of Santa Cruz, invited 12 of us to come and consider this as a possibility. He was thinking at the time about starting an institute to sequence the human genome. So we spent a day and a half and we debated this topic. And I came away with 2 really striking observations from the meeting. One was everyone agreed that it was technically feasible, although really very challenging. And it was interesting, the group was split 6-6. So when a few of us, Wally Gilbert, Charles Cantor, and I in '85 went into the biological community, it was with some surprise that I realized that 90% of the biologists were bitterly opposed to this. And it was really on doctrinaire grounds. Big science is bad science. It'll take money away from small science. And of course, the Genome Project's probably brought more money into biology than any other single project. And the other conviction was that there would not be any interesting information that came out of this, and you could never get good people to do the Genome Project, so it would be a mediocre effort and everything. And of course, these ideas, after a 5-year period of let me just point out that the bitterest opponent to the Human Genome Project was the National Institutes of Health, all the way up until the very end when it switched around because of a National Academy Committee meeting that gave validation to this whole effort. And of course, the only way the NIH could participate in this process was to set up a completely new institute whose object was to sequence the human genome, and it did a reasonably good job at that. So what was then very interesting was, again, a second example of a structure necessary for a new kind of idea. But what I came to realize in developing the sequencer, and we actually started much earlier than '84, was that to develop an instrument as complicated as that, we had to bring together really good chemists and really good engineers and people who knew a little bit of mathematics and computer science together with good molecular biologists. And in fact, once we'd assembled this team, we came up with the idea of the sequencer very, very fast. What this really impressed me with was the fact that why, in fact, should I have a very large lab? Because I wanted to make it cross-disciplinary. Why didn't we actually create a department whose faculty members were themselves these cross-disciplinary scientists? Why didn't we bring in all of these different flavors of science and have them in a department focused on biology? So I went to a different president of Caltech, Tom Everhart, and made this proposition. And he said, he was an engineer, he said, it sounds like a very reasonable idea. Can you persuade your colleagues? So the chemists and the engineers thought it was a great idea. The biologists vetoed it completely. And, you know, I never totally understood why. I can speculate, but I never understood why. And it was then that I made the hardest professional decision I've ever made, whether Caltech is a superb place, wonderful students and colleagues, but was I just going to do science the way I'd kind of done it before, or should I go some other place and set up a cross-disciplinary department? And I looked at the University of Washington, and in fact, Bill Gates made it possible then for me to think about doing creating a Department of Molecular Biotechnology, which was one of the first kind of integrated cross-disciplinary departments. And by integrated, I mean having all flavors of scientists, because you can argue there have been cross-disciplinary examples earlier on, but putting them all together, I think, was unique and very powerful. So this department started the field of proteomics. Two of the very fundamental first techniques for proteomics came out of it. We developed the inkjet technology there. Phil Green pioneered the key software for the Human Genome Project, both for assembly and for assessment of quality. We developed, Gerrit van den Eng developed a multi-parameter cell sorter that's kind of state-of-the-art today. And we had actually 2 of the 16 genome centers for interesting reasons there. But what happened was the dean who recruited me had said, look, if you'll come to the University of Washington, I'll give you the resources in the first 4 or 5 years to set up this cross-disciplinary biology department, and then I'll give you additional resources to build in systems biology, because as you'll see later, I've started thinking about systems biology much earlier than that. That dean, ironically enough, died in an avalanche in the Himalayas, and the next dean had really a very different point of view, and that was why in 2000 I decided to start the Institute for Systems Biology. And so let me give you a kind of grand field vision of how an engineer might think about systems biology with a simple analogy. Suppose that you wanted to figure out how a radio converted sound waves into radio waves. What might you do? Well, the first thing that you might do is a parts assessment. And if you think about biology, the last 40 years or so had been a parts assessment, looking at individual parts and to a certain extent trying to understand how they work, how they functioned in isolation. And then the Genome Project came along and gave us a relatively complete parts list. But that's only the beginning. The next thing you'd have to do is to assemble those components together in circuits and then understand individually and collectively how those circuits carried out this conversion process. And exactly the same is true in biology. As we'll see in a moment, information is handled by biological circuits, and the processing of that kind of information is absolutely fundamental to understanding the complexity of life. So what I'm going to give you is my own contemporary view and where I started first thinking about systems biology. And it was all driven by this idea that I became convinced of, that one of the big challenges in biology was in fact complexity. My lab was right next to that of Max Delbruck for many years at Caltech, and Max was really cynical about immunology, and he was really convinced that we were never really going to get anywhere with this one gene, one protein kind of approach. And I argued, well, we've done all these things with antibody diversity and everything, and he said, that's trivial. He said, the big issues are the immune response, tolerance, autoimmunity. We don't understand anything about those. And parenthetically, I'll say to a first approximation, that's exactly true today, too. I mean, there are more details, but there isn't really a global kind of understanding. So this idea of complexity started really driving me to think, in fact, that in contemporary systems biology, that there was this fourth very important type of information, that is systems, biological networks. And in the mid-'80s and late '80s, we didn't have really any tools for thinking about that in a very effective way. And I remember putting several grants into NSF and doing very poorly. Clearly, the reviewers didn't understand what I was trying to do. And, you know, I'm not sure I understood what I was trying to do then either. But the idea gained credence. We started using the term systems biology in the early 90s, and then I really started pushing for it at the University of Washington about 1996 or so. What is interesting, of course, is in the last 10 years there have been these grand convergences that have really made systems biology possible. So the Genome Project gave us the parts list that we needed. New computing hardware and, frankly, software have given us the ability to deal with information in ways we couldn't have done 15 or 20 years ago. And this, of course, is followed by new technologies that are really going to be revolutionary for miniaturizing, parallelizing, automating, and integrating information and data flow in really interesting ways. And we'll talk about one or two of those ways later. And, of course, these all converged around systems biology. And if you thought about systems approaches to disease, then you ended up with what we've come to call this P4 medicine. But central to this thinking was this old idea that we've had that biology really is an informational science. And let me say just a few words about biology as an informational science and give you as engineers a sense of what I mean by that. If you think about biological information, I would argue that there are really two types. One is the digital core of information present in your genomes. And of course, the second is the environmental information from outside your genomes that impinges on and modifies that input. The digital core, being digital, is ultimately completely knowable. And it's this core of certainty that really separates biology from all of the other scientific disciplines in a really interesting way. And I'll argue in 10 years we'll have the capacity to know every one of your genomes if you choose to do so. And from that, we're going to be successively capable of extracting really interesting types of information. So the essence of systems biology then was this ability to integrate digital information with environmental information and to look at that across the three time dimensions of life processes: evolution, development, and physiologic responses. The second point that I've already made is that, of course, I'm going to have to get this right, that information is processed by two complex kinds of structures. One are biological networks, and what they can do is capture the information, transmit the information, integrate and modulate it in really interesting ways, and then output it to the second structure, which are complex molecular machines that actually execute biology. And I'll just say that the interface between these two structures is not nearly as discrete as we thought in the past. But this is the essence of what we have to understand for systems biology. Here's a really complex circuit of 65 genes that mediates a certain part of sea urchin development. And what the person who pioneered this has done is gain such a deep understanding of how that circuitry controls this early part of sea urchin development that he can re-engineer the circuit in a rational way and create new predictable properties in the system. So he can create sea urchins with two guts rather than one gut and so forth. And the interesting idea we're going to discuss is in the future, we're going to use drugs to re-engineer disease-perturbed networks to make them either behave in a more normal fashion or to abrogate the more contentious disease-causing aspects of these particular networks. And the final point I'd make about biological information is that it is hierarchical and multi-scale in nature. You go from DNA to RNA to proteins to interactions and networks and cells and organs and organisms and all the way up to ecological environments. And the really important point to understand refers back again to the interaction of digital and environmental information. And the essence of what you have to be able to do in systems biology is pick a level of complexity at which you'd like to describe the phenotype, the output of biological complexity. Say it's the genes that, proteins that control the cell cycle. And it's important that you assess all of the previous levels of information all the way back to the digital core, and that you integrate them together. And by integration, I mean you capture the modifications that have arisen by environmental manipulations. And the environment impinges on every single one of those informational levels. So if you're to really understand systems, you have to be able to do global, comprehensive, and integrative studies, as we're going to talk about here. Systems biology has been around for more than 100 years, but what uniquely separates it, I think, today from before is one, we can do global studies. So we need to look at all genes or all messages or all proteins, and we need to because our biases about how biology works are always too naive, and only if you look at everything can you see the contextual interrelationships and so forth. Number two, you need to be able to carry out this integration we talked about. And of course, number three, we have to be able to capture the dynamically changing nature of the architectural structure of these entities that mediate biology. And those are all grand and central challenges in systems biology today. There is this virtuous cycle, if you'd like to call it, of biology driving the development of technology. But we'd add to that, once you have a new technology and you create data, that in turn has to drive the development of the appropriate computational and mathematical tools, not only for analyzing it, but for modeling it graphically and or mathematically, and then for creating this iterative cycle you heard in the introduction, where you test continually hypotheses until you bring formulated hypotheses into apposition with the implications of the experimental data. And all of this should be done on a global basis. It requires this cross-disciplinary culture. It requires all scientists learning the language of the other scientists. It requires the non-biologists learning biology, at least the biology they're interested in a very deep way, and it requires working together in teams. And these all three, in some ways, are antithetical, at least to classic academia. That's certainly starting to change now. What is interesting is if you think then about taking these systems approaches, you can apply them to almost any aspect of biology, and you can see how you really can transform these arenas. And what we're going to talk about is the transformation that's going to come about in healthcare through something that we've come to call systems medicine. And the basic idea is simple. Disease arises as a consequence of disease-perturbed networks, one or more in the affected organ. The disease-perturbed networks, genetically or environmentally, modify the output of information that they express, and they do so in a dynamically changing manner as the disease progresses. And it's the envelope of this altered information that encodes then the histopathology of the disease. And I'm going to show you an example of where we beginning to understand that in some detail. But even more, this picture of a systems view of disease gives us deep insights into new ways to do diagnostics, and that'll be one of the most important things I'll talk about tonight. And we're beginning to think about new ways that you can discover drug targets and ultimately carry out prevention. So I'm going to talk a little bit about prion infection in mice. It's the system we chose to study in detail for a lot of reasons that aren't very historically relevant, but being here in England, you understand that prion disease arises as a consequence of a protein that has been misfolded to become, quote, infectious. The infectious protein has two interesting properties. One, it can catalyze the conversion of normal prions to their infectious form. It's an autocatalytic kind of disease. Parenthetically, prions occur in a lot of different organs, and why the disease manifests itself primarily in the brain is an interesting and unsolved kind of question. The second thing that the infectious prion proteins, the second feature they have, is they're generally utterly resistant to any proteases that we know anything about. And what happens then when nerve cells take up these infectious proteins is they agglutinate, and this probably is a part of the mechanism that leads to evacuation and the death and destruction of the corresponding cells. So what we've done then is to take a series of inbred strains of mice and to take 10 different study time points across the disease period of induction. And this can range in some inbred strains from 60 days and in others 350 days. And then what we did was to look at each of those time points for 5 different inbred strains of mice, 3 congenic strains of mice, and 1 knockout. We looked at a transcriptome analysis of the diseased animals against their normal counterparts, asking simply what's changed, okay? And we're going to return to what's changed and how we, what the core of that is all about in just a few moments. But then what we did was to map this onto known networks that mediated major processes of prion disease. And then we correlated that with the histopathologic changes that occur in the brain across these same time points, and with the clinical signs. And here is one of the 6 networks that we've studied, one that deals with prion replication and accumulation. And it is a network that has of the order of 100 transcripts in it. Yellow means that the disease and the control are exactly the same in this particular case. And the lines around the subnetworks indicate transcripts or proteins that mediate certain subfunctions within the larger network function that you can define beautifully physiologically. So if we go to a later time point in this disease process, let me say in this animal that the death comes at about 23 weeks or so, you can begin to see the increasing levels of proteins in the diseased animals and a few decreasing. Red is up and green is down. And in fact, if we go along later, you can see an increase not only in changes within the networks, but the staining that you can see in the upper right-hand corner, that represents the accumulation of these infectious prion proteins. But the other thing you can see is that the clinical signs in these animals come only very late in the disease, and this is getting near the end when you've got the maximal changes in all of these network and subnetwork entities. Now, what is really interesting is we know a great deal about some aspects of the nature of this disease from really detailed histopathologic studies, and we can map beautifully network dynamics right into that histopathology. So we can explain a lot of things we already knew, okay? But the really interesting question, of course, is, what can we explain that we didn't know? And I'll just say that there are 6 networks that we've defined in exactly the same ways that we've defined the networks here. And we've been able to define in ways I'm not going to go into a core of about 900 genes that mediate this process of prion infection. And what we can say is about 1/3 of those genes explain these known networks, and 2/3 explain things we don't know about, including really interesting aspects of the innate immune response and things like that. So there are enormous opportunities then to expand our knowledge in a deep way, and this points out the enormous importance of global studies in systems biology, because we realize how little we understand about these complex biological processes. Now, if we take network slices at 3 different points in time in this prion infection, you can see again that there are changes in transcripts that occur 6 or 8 or even 10 weeks before you start to see clinical signs. So one of the really interesting questions to think about in terms of diagnostics is it possible some of those transcripts encode proteins which are secreted out into the blood, and we there could measure these very early events that antedate any of the clinical signs? And of course, that gets us into this whole business of blood protein diagnostics, and it's been a big business for the last 4 years in biology, and lots of money has been spent. And the really interesting point to make is we spent hundreds of millions of dollars on these projects, and we've gotten almost nothing out of it. So why is that? And the answer is really a very, very simple one. If we take 10 women that have ovarian cancer and compare their bloods using classic proteomics studies against 10 normal control women, we can really see a lot of markers that differentiate those 2 classes, okay? The problem comes when you take those same markers and you look at them in 10 other diseases in women. You find there are no correlations whatsoever. And the reason for that is those proteins are encoded in multiple organs, and they're subject to multiple environmental stimuli. So when you take one of these markers and use it in the blood of an unknown woman and it changes, you have no idea why that change has come about. And how can we get around this? A really simple idea. Let's figure out markers that only come from the organ that you're interested in. So we call these organ-specific markers, and we showed beautifully that you can use classic transcriptome analyses to get candidate RNAs that are specific. So, and this is an example in prostate cancer of something we did, but we've done the same thing for prion disease in the brain. And the really striking data are of these 900 core prion genes that we studied, 100 of them turned out to be primarily expressed in the brain when compared against 40 different transcriptomes of 40 different tissues in the mouse. So if we see changes there, we can say for sure those changes have actually occurred in the brain. And we have now a whole series of markers that allow us to do this really wonderful pre-symptomatic clinical diagnosis. This again is in mice, but you can show that the markers change of the order of 8 or 10 weeks before the clinical signs are onset in a statistically significant fashion. We've got multiple markers, and what's very interesting about these organ-specific markers is they're really highly conserved between mice and between humans. So at the 95 or greater % level for every organ-specific marker we've looked at, in mouse. Humans have a perfect ortholog. And what that says is we can transfer these diagnostic studies very quickly if we choose to do that, and we've not done that at this point. So the grand vision then is the following. You may have 50 or so organs and major cell types in your body. Each of those is secreting into blood a set of proteins that's organ-specific, and these constitute a molecular fingerprint, and the relative concentrations of the proteins within a fingerprint report the status of the organ because they reflect the network activity in that organ. So under normal conditions, you'll have one set of ratios for the brain-specific proteins. If you've got prion infection, the networks that have been perturbed will alter the concentration of their corresponding proteins, and you'll see a shift in a subset of the proteins within there in a way that is absolutely characteristic of the disease. So there are two really interesting points that come out of this. In the future, diagnostic markers won't be this point-to-point, here's a marker for prostate cancer, but it's going to be, here are 50 markers that will give you every single disease in the prostate or in the brain or in whatever. So it's a fundamental paradigm change. And of course, the other point that's really interesting is if we have 50 tissues and if we need of the order of 50 proteins for each of those organ-specific fingerprints, and we do for a reason I'm going to get into in just a moment, how are we going to go about figuring out how to measure all of those proteins from a fraction of a droplet of blood? You have to go to microfluidics and nanotechnology. But why is it important to be able to do 50? And you need to have an enormous dynamic range in the proteins that you're looking at because little organs have little fingerprints with regard to concentration, and big organs have very big fingerprints with regard to concentration. So the two things we'd like to be able to do then are these pattern correlations that distinguish health from disease, and if disease, which disease. But second, we'd like to be able, from the blood, to actually be able to infer the nature of the networks that have been perturbed in that organ. And with 50 markers, we might just barely be on the edge of being able to do that, and it's really a hard and interesting computational problem. But if we can do it, you can do early diagnosis, you can stratify disease, you can assess the stage of progression, you can look at response to therapy, you can do many, many different things. And in fact, equally important, these blood molecular fingerprints are going to let us do human biology in a way we never have been able to do it before because they are the ultimate measure of this integration of digital and environmental information, if you think about it. We've not ever had that measuring capacity in the past. So let me talk about two technologies that I think are really going to be transformational in this regard, and one is microfluidics and nanotechnology for these blood-specific organs, and the second is the high-throughput DNA sequencing approaches. And really what we'd like to be able to do is make 2,500 measurements from a fraction of a droplet of blood in a highly precise and a specific manner very rapidly, and to do it in a massively parallel way. I mean, if this type of diagnostics works out in the future, you can imagine that you'll be doing hundreds of millions of diagnostic tests on corresponding numbers of patients 2 or 3 times a year. So you have to have the throughput to be able to handle that new kind of medicine. A collaboration we set up with Jim Heath about 4 years ago at Caltech. Jim is a really spectacular young chemist interested in nanotechnology, has progressed to the point where we've developed a new type of protein chip that basically allows us to take protein capture agents, which are now antibodies, and couple to each specific capture agent a 20-mer DNA oligonucleotide, and then to place on a chip to address that capture agent the complementary oligonucleotide. So if we take N agents, and you can have 20 capture agents, or you could have 2,000 capture agents. The limitation here is one thing. It's where you can get your capture agents, and this is an enormous problem. And in the future, it isn't going to be antibodies. I'll guarantee you that. There are some very exciting new chemistries for looking at that. So the idea is you take your various uniquely labeled capture antibodies and their addresses, and you pour the antibodies over the chip, and they self-assemble in their appropriate addresses. And this gives you a fantastic ability to deal with what plagues protein chips, which is biofouling. And the reason we can deal with biofouling so well is this beautiful negative charge of the DNA molecule. Just an accidental kind of thing that turned out to be fantastic. And this allows us to get enormously good signal-to-noise responses, which we need if we're going to have this very large dynamic range that we talked about. Jim has done some really brilliant work on microfluidic engineering of the front end of the input system for this analysis, where we can beautifully, using just physical chemistry, separate cells from 300 nanoliters of plasma and then pass the plasma over the DNA chips, and we're now barcoding the various protein capture agents in various ways. And the really important thing about these chips is they have two absolutely critical features. One is their sensitivity is low femtomole, high attomole now, and we can actually use this technology to do digital counting of protein molecules. We can look even at individual protein molecules if we wish. We're not doing that on a routine basis. Number two, it has a dynamic range that spans about 10 to the 8th. And as you know, most measuring technologies are lucky if they have 10 to the 2 or 10 to the 3. And that's really important because, again, in the blood, we want to be able to encompass that dynamic range that includes proteins from the large organs as well as proteins from the smaller organs. So let me turn to another technology, and I'm only going to describe it, and that is single molecule analysis. And you may know that there are a lot of different technologies for very high throughput DNA sequencing out there. And the one that I think really is going to be the big winner is one that's being developed by a company called Helicos in Boston, because it does single-strand DNA sequencing. And what that lets you do are two things none of the others can do. One is there's no preparation necessary for that DNA. You can take a small number of white blood cells and get the DNA and put it on the chip. And two, you can think about a parallelization that'll give you 1 billion strands from your DNA that could be sequenced simultaneously. And if you can do that to 30 to 50 base pairs, you've got enough information to get maternal and paternal haplotypes. And of course, what this opens up in really exciting ways, and you know what other approaches are trying to push forward too, is the personalized genome project. And I would argue we ought to easily have, within 8 to 10 years, the ability to sequence your entire genome for well under $1,000, to do it rapidly, to do it relatively accurately, and in a very efficient manner. The other thing that is really interesting is we're pushing a new idea at the Institute called systems genetics. And the idea for systems genetics is classical genetics has attempted to take genetic variation in the genome and to correlate that with phenotype and to study those correlations by watching the segregation of information in matings. And one of the interesting ideas is, if you think about connecting digital information to phenotypic information through networks, then there are really interesting ways that you can think about doing single-cell analyses that may let you skip a lot of genetics. And one thing that we're beginning to think about doing with Steve Quake is to be able to take 1,000 macrophages from your blood, park them in 1,000 different little chambers, and then perturb those macrophages with 100 different environmental agents that will let us interrogate different aspects of the network behavior of those genes and capture that information. So you have the ability in a very rapid fashion to begin accumulating the information you need, again, for connecting genotype with phenotype through network behavior. And of course, all of this takes us, that is the systems approach to disease, the new technologies, a lot of computational tools that we really haven't talked about at all here, point toward the idea that this P4 medicine, as we call it, is really going to come to the fore in the next 5 to 20 years. And in fact, I think the first two Ps, personalized and predictive, are really going to come very quickly. And key tools are going to be a lot of the things we've talked about here. So 10 years in the future, I can well envision that many of us will have our genomes done, and from that we'll be in a position to begin making probabilistic assessments of future health histories. You have a 80% chance of prostate cancer by the time you're 60 and the like. And at the same time, I think we'll have these simple handheld devices that will prick your thumb and make 2,500 measurements and send that via wireless to a server, and that information will be processed, and you and your physician will get back an email that says, you're fine, do it again in 6 months, or go see your rheumatologist, whatever is appropriate. From the point of view of personalized medicine, everything I've talked about really is personalized, and it's based on a medicine that takes into account the unique individuality of the fact that each of us differs on average from our neighbors' genomes by 6 million nucleotides, and hence we're susceptible to differing combinations of disease. One of the really interesting aspects of personalized medicine in the future is the patient is going to become the control for assessing their own disease, because we'll have enormous longitudinal information from these blood organ-specific fingerprints. We'll be able to follow things in a very, very precise way. And what the absolute levels of proteins are in those fingerprints doesn't matter at all. All we care about are those subtractive analyses. And that's a fundamental new way of thinking about personalized medicine. There are lots of other things we could talk about. Prevention starts, of course, with using the fact that we're going to be able to choose targets in an entirely different way, because we're going to think about how we can use drugs to change network behavior, to push it toward disease network behavior toward a more normal fashion. And of course, one of the really interesting ideas is, will we be able in the future to take that individual with an 80% chance of ovarian cancer by the time she's 50 and say to her, here are a series of drugs which, if you start taking them when you're 35, will prevent that network from becoming perturbed and will reduce the probability of that disease to 2%. And that's what I think preventive medicine is going to mean in the future. The participatory came out of a discussion with Larry Page. Larry is big about information freeing individuals, and he really loved the idea that in the future, if we could deal with the education problem, and that's twofold, the education of physicians, and that's probably the harder of the two problems, and the education of patients, then you may indeed give them the information they need to play a much bigger role in charting their own future health history. So that's P4 medicine, and that's where I see things going. Now let me talk about two really striking implications from P4 medicine. I think the technologies we've talked about are going to lead to a digitalization of medicine, and it turns out that digitalization of medicine is really important, because Boolean analyses of yes or no give you absolute certainty on specification of patients that will respond to good drugs, all these kinds of things. And I would argue the ability to extract information from a single molecule, information from a single cell, or relevant information from a single individual is going to be the basis for this digitization. And I would argue the revolution for society will be far greater than that for IT or communication, in the sense that the most profound implication coming from this is that it will reverse this ever-escalating cost of medical care, I think in a very, very sharp way, and start it on a downward trend that will put us at one time in a position where we can think about exporting P4 medicine to the developing world. That is, P4 medicine will become the core of global medicine in the future. And I'm trying to convince Bill Gates of this, but he's a pragmatic guy that wants a little more proof than we have at this point in time. So that's one implication. Now the second implication has to do with what this is going to do to the healthcare industry. So if you look at all of these different sectors of the healthcare industry, drug and biotech and IT healthcare and payer provider insurance and so forth, you can argue that within 10 years they'll all have to rewrite in a very fundamental way their business plans. And in fact, the really interesting question which I posed earlier is, can old organizations respond to really new ideas and change? And I think for big pharma, many of them mouth the dogma of systems biology. Very few are even remotely close to effectively practicing it. So I think that the jury is out on what exactly is going to happen. And I think wherever there is this enormous these disruptive technologies that come in, there are enormous opportunities for new, young, agile companies to come in and really become important players. And perhaps in some ways, nowhere is that truer than with IT for healthcare. And I think the IT that most people think about for healthcare is this kind of trivial, important but trivial digitization of medical records, but it goes far beyond that. I mean, for example, IT is going to play a really critical and central role in this education of physicians and education of patients in a way they've never even started to think about. And of course, the final point I would make is medical schools are another business that ought to really be rewriting their business plan in very fundamental ways, and how you can persuade good medical schools to make the kind of changes in any more than a trivial kind of incremental fashion, I think is a fascinating question, and I've had a lot of experience in this regard, which I'm not going to get into here. But what I would like to do is to close with one point. In addition to ISB being interested in systems approaches to things and developing new technologies and new computational tools, one of the things that we are committed to is remaining small and cohesive and focused and interactive. And that means if we're to take on really big problems, we have to create strategic partnerships. So let me tell you how we're thinking about these strategic partnerships. And I would argue there are a whole series of big problems in biology that could benefit by the kind of thinking I'm going to talk about for P4 medicine. And bioenergy is absolutely a classic kind of example. I can sit down and write 10 milestones for P4 medicine, doesn't matter what they are. And my deep conviction is if we solved all 10 of those milestones, technically we would have solved the problem of P4 medicine, okay? So that's point number one. Point number two is all of that probably would come to naught unless we dealt with the ways that P4 medicine is going to impact on society, and those are going to be equally profound, equally disruptive, and probably much harder to deal with in many ways. So what we're thinking about doing is creating a consortium where we're trying to begin to bring in both academic partners, but as well industrial partners. And there we're focused on getting one really leading edge partner from each of these major arenas in the healthcare industry that will, together with us, take on attacking some of these fundamental milestones. So for some, those would be the technical milestones, and for others, those might be the social kind of milestones. And we have PricewaterhouseCoopers helping us in a major way, and I think by the end of the year, we'll have a major business plan and our first 4 or 5 partners in this whole endeavor. And the idea is really an interesting one. With a focused effort by committed and excellent scientists, I really think we can enormously speed up the realization of P4 medicine. And the people who come into the consortium will gain not only by the enormous benefits they'll get for solving problems relevant to their own fundamental interests, but they'll gain enormously from seeing how other non-competing industries are solving their kinds of problems. And this is the kind of business plan that we're trying to put together at this point in time. So one of the fundamental, essential core needs for this thing is to have at least one medical school that's willing to refashion themselves in the terms of systems medicine. And I think the way to do that is to set up an institute of systems medicine that has lots of resources and lots of flexibility and freedom to operate in the right way and not be constrained by bureaucracy. And my interesting experience, as I related to some who were in the audience earlier, is I've gone to 2 first-class medical schools in the top 5. There's no way they're going to do this because of the arrogance of excellence, okay? I mean, these are schools that are confident they're the best. Why should they change? And they've got really smart people at the administrative and at the scientific level who have other agendas, and they see fundraising as a zero-sum game. If you get a lot of money for an institute, I'm not going to get it for my translational institute or whatever it is. So this, but we found one that's not in the top 10 who would like to get in the top 10, and this is probably the best way they have of getting in the top 10. You know, this is exactly what happened in 1918 when the Carnegie Institute made this very now famous report called the Flexner Report. And the Flexner Report simply said, the future of American medicine is going to be the integration of basic science with clinical practice. And Hopkins leaped on this, hired 4 guys that really made that work, and they went from being a mid-level trade school up to the very top. And I think these are the kinds of opportunities that avail themselves. The other point I would make is there are abroad some incredible individuals with complementary skills and technologies and strategies that we're beginning to explore partnerships on an international basis. And what we're actually thinking of doing is setting up clones of ISB, philosophic clones of ISB, in a number of different places. And we're furthest along right now with one of the countries in Asia. And I must say, I've tried this in 3 other countries now and failed, and I learned a lot of lessons with each failure, so I think I know, I can identify very quickly what's going to work and what isn't going to work. And I'll say in Asia, it's really interesting. I think most of the countries there haven't got a ghost of a chance of doing this for a lot of interesting reasons. I think there are 2 that really could push these kinds of ideas, and I'll bet they wouldn't be the 2 you might think of. But we are very close to putting together another kind of partnership. And what excites me about this is I think for big problems, there is going to be a globalization of science. It's very much akin to what, to the flat world that probably many of you've read. And the idea there was, Tom talked about the whole process of the globalization of the economy. And I think there will here be a globalization. And the benefits will not only be to draw in strategic partners, they will not only be to gain access to really talented kinds of people, but they will, in the short term anyway, be to open up brand new, very large funding opportunities that most other people haven't thought about. And in the US anyway, if you think about the needs of P4 medicine, one of the big needs is a lot of resources because we know how to do a lot of things now. It's just going to take a lot of money, and it isn't going to come from the government. Between the war and the tax cuts, discretionary money in the US is in real disarray. The real cost of the war is only next year going to come on the budget, and we're going to see absolutely disastrous consequences. So we have to think about other ways of creating the kind of resources to do the science that we want to. And I would argue that those who ride this crest of the globalization of science in this good sense, looking for excellence and focusing on deep and fundamental kinds of questions, really have an opportunity to move their agendas forward. So that is what I had to say. Let me just make one more comment. The white slide was my closing New Yorker cartoon. And it was a guy looking down at his cat in the cat box and saying, "How many times have I told you not to think outside the box?" And what we have to do is figure out how to think outside the box. And we aren't very good at doing it. And the other really interesting question is, how can we teach our students to think outside the box? That's, I think, really the fundamental question. So I'm open to questions, if there are any questions.
Thank you very much indeed. Yes, we have one immediately. Could
Sure.
I just say before we start, because this is being recorded, could you please say your name and your affiliation before you ask the question? Thank you.
My name's Elizabeth Bell. I work for... Hello, can you hear me? Yeah, my name's Elizabeth Bell. I work for the Physiological Society, which of course... The Physiological Society in London, which I think is perhaps the oldest society in this area in the world, and has been sort of worried about physiology, medicine, biology since sort of middle of the 19th century. So first I'd like to say that in the last few years we've really got interested in this whole issue of systems biology, and we're 100% behind your report and the sort of things that you're doing, because some of the very eminent people in our society really believe that this is the way forward. So, you know, please talk to us about this area. I was also very...
Let me just say, you guys started it, right? The physiologists did,
we're natural
really.
innovators, you know, we can't help
yeah.
it. But I was also struck by what you were saying about the importance of building links with the developing world, because something else that our society is now sort of working on is this issue of capacity building in science, technology in the developing world, because a lot of the big aid agencies have now got interested in this area. And I went to a very interesting World Bank forum in February of this year in Washington, where they were basically scratching their heads about what they could do in this area. And it strikes me that systems biology, working with developing countries to help revolutionise their healthcare systems and all the research systems things that underpin that, just might be the big idea that they're looking for. So maybe we can talk.
So I can talk about that. I've actually talked with several people about this possibility. And my idea is the following. I think with sufficient outside support, you could set up an institute to do systems biology in ex-African country, okay? And with the resources, you could recruit really first-rate, top-flight talent that is good, but wants to do science and would love to go back and be able to do that in Africa. And I think from that, then you could begin to disseminate seeds both for training and for doing research that could be very powerful. Now, what is totally clear is those governments in no way have the resources to be able to do that. So this is a project I'm really hoping to persuade Bill Gates to take on seriously. And I have to say, if they'd spent money on this rather than on the AIDS vaccine, we'd be infinitely further ahead than we are right now. And that's because, you know, we don't understand how to create cellular immunity in vaccines. So picking your shots like this, I think, could be revolutionary. And not only if you set up a really good institute would it help the country, but it would help the region. It could begin to build a network of excellence and so forth. So I think that's really a terrific kind of idea. But it would take sufficient funds that you'd be able to really recruit talented people who could do this very effectively.
Yes, sir.
Hello, my name's Nicholas Beale from SciTeb. Dennis Noble wrote a wonderful book called The Music of Life and has been giving talks about what you might call the philosophical implications of systems biology and how the idea of reducing everything to the genes as opposed to understanding that the genome is a very important aspect of what goes on, needs to be challenged. And he's proposing that there is no single privileged level of causality in systems biology. You seem to be more emphasizing the genes as the sort of the fundamental, but I wonder if you care to comment on, genetic reductionism as opposed to systems having to look at every level of the system?
Sure. Well, I tried to make that in this discussion of biological information, that there are all of these hierarchies, and you don't understand systems unless you capture them all and understand that interface, again, between the digital genome and the environment. So it is that blend together that really I see as the essence of systems biology. So I agree with them absolutely 100%. I mean, we aren't, for the most case, going to find genes for X, okay? And in fact, I would argue that the greatest example of really spending a lot of money on genetics and not getting very far are these genome-wide association studies that have been done in a number of cases now, and you spend a gazillion bucks and maybe you come up with one gene, and you know already that almost certainly complex genetic diseases might be the combinatorial effect of 6 or 8 mutant genes that can be drawn from a pool of 30 or 40, and you get one of those genes. So where does that put you in terms of understanding the disease? Puts you nowhere. And the real problem is the signal-to-noise issues in genome-wide association studies. They're never, ever going to get to the point where... So I think with personalized genomes and the ability to integrate genetics with the environment, as I described, I think this is where we're going to get the right signal-to-noise ratios that will let us begin to penetrate the true complexity of these complex diseases and so forth. So I agree completely with him. So if I left any impression in the audience that I'm a genetic determinism person, that is not correct. But genetics is really important, no question.
Yes, the microphone there, please.
John Lowe, Sir Henry Royce Memorial Foundation. What mechanisms do you see to deal with the societal issues, for example, ageing, for example, revised healthcares, and to which you make reference? Seems to me that they will swamp the efforts to look at the biomedical situation.
Well, I think you have to look at each of those individually. So my own conviction is that if you look at what we can do in systems medicine, if you look at actually some of the really interesting studies that are coming out on degenerative neurologic diseases and in fact on aging itself, beginning to understand some of these potentially basic mechanisms and so forth, I think we can see in the next 20 to 30 years a real extension of the productive lifespan of individuals. I think there's no question about that. But I'll tell you, the really deep and fundamental issue is what happens in the last year. That's when we spend, 50% of the healthcare we spend the rest of our life. And how is this kind of medicine going to help us deal with that? And the answer is, it isn't. That turns out to be probably a societal question and not a scientific question, because, the last year is kind of massive systems breakdown, and I don't know, maybe we'll be able to deal with that, but I doubt it. So that is, it's a fair issue and an important issue, but the other thing that I would say is most societies don't treat older people very well. And if you have older people that are creative and productive and alert into their 70s, 80s, 90s, it would be a shame if society didn't take advantage of those enormous resources. On the other hand, what happens to the younger people that nature has removed us older folks for, and how society deals with those? I mean, I think there are a whole set of new problems that are going to be absolutely fascinating.
I just wondered if you knew of any mechanisms by which those deep issues are being addressed. Maybe at UN level or at Bill Gates. Is anybody that you know of attempting to steer societies towards proper consideration of those issues?
So I would say no, other than those of us who are kind of aware of them and can articulate them in discussions like this. But I don't know of any really organized groups that are trying to do that. I will say that earlier this year, I got together some really interesting people from Harvard, from Yale, from Princeton, and a couple of other places to sit down and spend a whole day projecting what they saw as the major impacts of P4 medicine on society. And that was an incredibly interesting kind of discussion, but it really just laid the framework for where do we go from here.
So just take one back first, yes? Yes, please. Microphone back.
All the way back,
Back,
yeah.
yes, we're trying to spread the questions around a bit.
Hi there, Daniel Gooch from the Department of Health in London. Excellent talk, really enjoyed it. What plans do you have for an ISB clone in London? We need one.
Well, Imperial College might say that they do have one. So, you know, it's really interesting. About 5 years ago, I went and spent a bunch of time at the Karolinska Institute trying to persuade them to set up an institute to do systems biology. And I went to the Pasteur Institute and did the same and got very little traction. And I mean, it wasn't, in all cases, there are always people really interested in this, but there are a lot of people who say, we're already doing all of that, or we don't need it, or we want to go in other kinds of directions. So, you know, it's hard to build, it's like the medical school, it's hard to change really good institutions. They don't have the motivation for change. So, so they do it internally, and some can do it efficiently, and most it's an awful lot of the systems biology efforts I've seen are frankly more of the same. They aren't really, and maybe they'll learn how to do them, and maybe they won't. I mean, again, I will say in big academic organizations, there are a lot of barriers to doing systems biology right, and I can give you chapter and verse on 5 or 6 or 10 of these things that I ran into, and maybe it's different here, I don't know.
Yes, Ian.
Thank you. Ian Sutherland, Director of Brunel Institute for Bioengineering. Personalized medicine is going to lead, obviously, to revolutionary changes in bioprocessing and biomanufacturing. With versatile, small footprint manufacturing processes. Do you think we're ready for this? That's one question. But also, you mentioned Asia and the interest in traditional Chinese medicines, where they take more of a sort of holistic, systemic approach.
Right.
Do you have a comment on that also?
Sure. Well, as to your first question, I don't think society is ever ready for any revolutionary change. So it's always hard to make change, and I think the change itself, if it's successful, creates the opportunities that mandate and drive that kind of change. So whether society is ready or not for P4 or personalized medicine, I think it's pretty inevitable. And you can argue about time scales, and you know, look, what are the time scales? There are two factors that determine the time scales. One is technology, and I would say the relevant key technologies are in exponentially changing Moore's law kind of mode right now. And the second is funding, and that's again more problematic, and that's why I talked at the end about how we would approach it. So I just went about 6 weeks ago to the first international meeting of traditional Chinese medicine, and I was asked to give the keynote talk on how systems biology could help and inform traditional Chinese medicine. And it's really going to be a hard conversion. and I'll tell you why it is. Almost all the people at this meeting did not speak English. And if the translations I knew something about from American to Chinese were as bad as the ones from Chinese to English, I don't think those people got very much out of this at all. And there are a few real pioneers, but I think the core practicing people are extremely conservative and very much steeped in ritual. I will say, to my surprise, after we got through a few preliminary talks, there was an enormously entertaining and engaged discussion. But again, I think it was it was among mostly the cognoscenti. It didn't really draw deeply into the traditional Chinese medicine. But I would argue, and I argued then, there are enormous opportunities for cross-fertilization in very interesting ways. But we'd each have to learn a lot about what the other can do for us. So we'd have to create this true cross-disciplinary kind of culture. And whether that'll happen or not, I don't know. I was just struck by this language barrier that really separated the two groups primarily. And yeah, those are the two questions you asked. Yeah.
I think we'll make this the last question, if you could, please.
Hello again, it's Liz Bell from the Physiological Society. Just quick comments on what other people have said. Firstly, the whole issue of working with, say, traditional Chinese medicine is, I can imagine there was a massive language barrier in terms of going to that country, talk to people in that country about it. But there are a lot of those sort of practitioners working in the West who do speak English, so it might be more productive if you went to speak to practitioners in England and America on that. Also, I was just struck about the question about how society might be able to adapt to radically increased lifespans. And I just like, I'm quite optimistic about this because our societies have already adapted to radically increased lifespans in a very short period of our history. In the last 100 or 200 years, our lifespans have increased massively.
Sure.
And I don't think we're going to have much trouble adapting, to be honest.
I hope you're right. Yeah.
So, could I now ask you, in thanking Leroy Hood for this wonderful talk this evening, we've seen complex problems really do need very original thought, and we've had that in plenty this evening. Thank you very much indeed.